Our next speaker is well known to me, Jin He, who was in my lab. When did you start, Jin? I can't remember. A long time ago.
2000.
You got your PhD in physics, didn't you?
Yes.
Before I moved over here, right?
Yeah, and no, after.
Oh, did you? Okay,
Yeah,
all right,
I
okay.
have,
So Jim was right at the start of biodesign, working on scanning probe microscopy, and he's done a lot of very original things at Florida International University. An interesting side note here, Nongjin Tao was the star of their department there, and of course we recruited Nongjin Tao away here, and regrettably he's not with us anymore. So FIU went out and recruited Jin, so he had big shoes to step into. So Jin, over to you. Thank you.
Thank you. Hello, everyone. It's my honor to be here as a Biodesign alumni and to discuss some of my research and also to show you that how biodesign shaped my research career. So here a brief history as Dr. Lindsay just mentioned. I came to SU in 2000 and to the physics department. So 2001, that time the summertime, then I checked several labs, and then I decided to join Lindsay's group, and I actually start doing some research in Lindsay's lab that time with a graduate student, Xiaodong Cui. So then 2004, we moved to the building A, right? That's only building of biodesign at that time. Actually, the same time the trend moved to building A, we actually in the same color. And also so several other groups there, Neil Woodbury and the John Chapu and also the Yan House group. So all there in the same place. So in 2005, I graduated from physics department, and then I continue to stay at the biodesign and postdoc, and then later assistant research professor. So I stayed at ASU for 11 years. So until 2011, then I left biodesign and I joined the physics department of FIU, which is in Miami. So then I stayed at FIU for like about 13 years. So my career actually pretty simple. So most time just ASU and then FIU. So here I show the pictures, both the Lindsay and the Longjian Tao, because I want to mention that both of them are in my PhD committee. And I really feel fortunate to have them as my advisors and mentors, that they teach me a lot, that they really shape my current research. So because the research is so my career pretty simple. So I will try here to show a lot of examples for research. So my research journey, so how the ASU, my learning and the trainings and ASU, how that shape my current research. So that can show some impact. So I will give you several examples, what I did and ASU, and then what I do and FIU. So we'll see the collections, you will see how ASU impact my current research. So in general, my research in like three like major topics. So one is we try to use nanotechnology, try to construct nano conduit to confine and modulate electron, ion, and molecule transport. So because my background in physics, so we like to study more fundamentally like electron transport, then later ion transport, then we apply to the molecule level. So as a physicist, we also try to understand the mechanism. So try to find some new phenomena and try to understand the mechanism for the electron and ion transport. So after that, then because in biodesign, we also try to apply what we learned and the nanoscale for the electron and ion transport for applications. So we try to measure molecules, especially individually using the electronic signal and the ionic signal, especially in the physiological condition. So I will start with electron transport. So for electronics, so that time like 2000, right? So that time electronics still the major thing. And the electron transport is most fundamental problem to have efficient electronic devices. Sterling's lab, that time they're interested to study electron transport and the molecule level. So at that time, the field is try to see if we can use molecule to replace the silicon material as new nanomaterial. Then we can build a functional device based on the molecule. That can be much cheaper and then scale much smaller. So here I show you the some the fundamental things about electron transport. So one thing is electron is so small, so we cannot use classical physics to study. Ohm's law will not apply at the nanoscale level, so we need to use quantum approach, like a Landau approach. Think about if we study electron transport through gold atom wire. For that very small atomic level structure, there's no electron scattering. So electron transport mostly defined by the quantum mechanics. So the available quantum channels, right? So here shows the data that we can see electron transport through the 1 step, 2 steps, 3 steps, that we call quantum steps, which can be derived here. So if you break the junction, then if we break the molecule into the junction, then you have molecule orbitals available, and then you apply voltage. You can drive the electron go through. Either if the molecule pretty small, you can get a tunneling current, right? You can also then doing a lot of stuff. So over the years, we studied all kinds of molecule. So Josh still doing a lot of fantastic stuff for the molecule, so study electron transport through molecule. So that's it, like about 2001, that time, right? So the major question that time is how to reliably wire the molecule into the metal junction. So that's technically very challenging. And so in 2001, Xiaodong in Sterling's lab that developed a method using conducting FM, right? They press the conductive tip to a nanoparticle that self-assembled to the molecule. Then they can achieve reliable conductance from molecule. And at that time for me, we want to think about applications. So I go to the clean room. So we try to fabricate nano devices. Then we can bridge the molecule into the junction. So so I make a lot of devices in the clean room in the engineer school. And then later we do some electrochemical deposition. So that is supervised by Long Jiantao. So we can make a nano gap, and we did a lot of measurement, but the data not very reliable. Right? Sometimes you get a good signal, and the other time you try to repeat, you cannot. So I struggled for some time. I make a mock device, but cannot get reliable result. And in 2003, Long Jiantao's group, so they developed another very elegant method, similar to conducting FM, but use simpler, just gold tip. Then they drive to the gold substrate, then stretch out, so we call the STM break junction. So in that way, you can very quickly measure the electron transport through molecule. So that method now becomes the mainstream, the main method in this field. And so after like more than 20 years, so many people still using this method. So this kind of method also saved me for my PhD studies. Then I go to Long Jian's lab and try to learn this. We recopy the method, and then we use this method. studied several molecules. Later, so this shows some later when I at FIU, then I using what I learned before. We also make girdle tips and we modify with molecule. Then we put the nanoparticle on the surface. So in this way, we also form a nanoparticle and nanoelectrode junction. We can study molecules. So this is more for the like a Raman spectroscopy study, but it's very, because of the tip, so they can enhance a lot the Raman signal. And this, you can see that actually I learned a lot from either conducting FM and the STM method to develop the new method. So after we learned the method developed in the Longgen Taos group, so one way we want to try, because Stuart always interested to study molecule in the electrolyte. So at that time for molecular electronics, most of studies to study molecule in dry condition or in vacuum, right? But a lot of molecules, especially biological molecule, they function well in ionic solution. So then we start with electrochemistry. So we make this EC STM. So the tip is insulated. Then we can control the potential on the tip, on the substrate, then also the double layer surrounding the molecule. Then we can modulate the molecule conductance, because so for example, this molecule is aniline oligomer, which can be oxidized and then they become the conductor. So the data here show that the red curve, the conductance change a few times if the molecule being charged, so they become conductive. So what we learned from that early experiment, that electrochemistry really is powerful, that we can play with electron transport, with ion and the molecule, so we can modulate effectively electron transport, and also the ion concentration surrounding the molecule is important. So they provided the correct electrostatic environment and the modulate electric field gradient, so we can change the molecule and also the electron transport. So later, I also try to utilize electrochemistry in my study. Then I also continue to study redox molecule. Now we add the light, so we excite the molecule by shining the laser. So one good thing about the metal electron like gold, so we call the plasmodic metal. So they can have surface plasma, which is a collective oscillation of the conducting electron. So with that surface plasma, we can also generate the electron hole. And those we call hot electron, they can transfer to the molecule. Then they can oxidize or reduce the molecule depending on the transfer by electron or hole. So this provides another way that we can change the charge state of the molecule. So we one of the work we play with the using the plasma, and then we use electropotential to oxidize the molecule, like from NTP to the DMAB to the ATP. Then we can then reduce back by applying electropotential. Then we can go back. So in this process, we can reversibly oxidize or reduce the molecule by carefully balancing the strength of the plasma and also the electropotential. And also here actually we use copper electrode. Copper is another plasmodic material, but they also have more reductive power compared with the gold. So actually here we can successfully reduce the molecule from NTP to the DMAB because of the presence of copper. If we use gold, we can only see the reduction from DMAB to ATP. So that also, if you can play the different properties of electrode and the plasma and the electropotential, we can control the charge state of molecule. So this may need to some application. One is for the, we can do the carefully control of the reaction, and also we have a better way to control the charge state of molecule. You can also then do the electron transport, right, if you can control their charge state really well. So now I will change gear to the ion transport. So after we play electrochemistry, so we really interested in the iron. And also talk about the ion transport and the nanoscale, right? So one famous example will be ion channels that exist widely in our body. So here shows some like a sodium, cation, potassium channels, right? So they're on the cell membrane. So those ion channels, they regulate the ion transport. So they can change the ion concentration across the membrane. So you can polarize or depolarize the membrane. So with that change of concentration gradient across the membrane, you can change the, one is the membrane potential, which at the rest, or maybe action potential when they are transiently changed. So then they can regulate the cell activity and also the cell signaling between cells. And also for the iron, then you can also develop like ionics. They're similar to the electronics, right? So integrated iron circuit. So they can have new functions. And also combine ionics with electronics, there will be like iontronics. So here show that we try to develop different nanostructures that we can control the ion transport. So at ASU, we developed a carbon nanotube iron channel and a graphene nanopore. And FIU, then we start some simple approaches. We put in the glass capillary tubes. Then we can form this very sharp needle. And the tip, you will have a very small hole. That's not as small as carbon tube or graphene, but typically can get down to like a 10 nanometer and above. So that's still, because it's so easy and cheap to play with, then we spend a lot of time trying to use the nanopipette. So when we have that device, right, so how to measure the ionic signal? In general, will be similar like electron transport, but for iron, it's bigger than electron, right, and heavier. So actually the classical physics can understand electron transport. We don't need the quantum mechanics, so that part is simpler. Like we can use the electrostatics, statistical mechanics, and the fluid dynamics. Then we can calculate the ion transport through the catalytic nanostructure, right, bridge to reservoirs. Then you apply the voltage, you get the current. And also you can apply some electrostatic gating to modulate the transport. So here just use some example for how we calculate. Normally 3 terms, the concentration gradient, the electric force you applied, and also the fluid movement, right? And here one example that we do an ionic circuit by using the carbon tube and ASU. So we make that device, we have a gate here, so we apply the gating, we can get the ionic transistor. So there's a P-type ionic transistor. So similar like electronic transistor. So the gating effect not as good as electronic circuit, but they also have certain functions. Okay, another example that we applied iron current that we use for imaging. So we combine with scanning probe microscope. So you see that we can get a very pretty nice the imagings for the cell membrane, then for the tissue. So I'll not go to the details because of time. Other things we also study, we put the membrane on the tip, so we can study the peptide, how they form the ion channels on the membrane surface by simply measuring the ionic current. So because they can form nanoscale lipid, so they are much stable and they can last for a longer time. So here we show example. The typical signal we got that we can just modulate the voltage. So with that negative potential, we can insert the peptide into the membrane, and then you see those signals. And when you change the voltage back to 0, so they will be kicked out. So if you zoom in the signal, you will see actually all this stepwise change. So that because of the peptide, they insert into the membrane one by one. So we can, if we do the statistic for a long time, you will get all the peaks, conduction peaks, and they can match with the very simple geometric model for the size that you have like 5, 6, 7 peptide insert there. So that there's some deviation when the number of peptide increase. We think that due to some dynamics of the peptide, when the pore is become bigger and structure become bigger. So for the molecule part, because of time, I will just only mention a little bit that we try to apply then what we learned when we try to study electron transport, ion transport, then we apply to the molecule. Because that we can do single molecule detection, we can do label-free and real-time measurement, right, compared with optical method. So over years, we developed different methods. So I will talk about here, like one is we let the molecule go through an ion channel, so we can measure the ionic current, or we can use embedded metal electrode, you can measure the electron transport. So like this part, the Sterlinsis group, I think, is still working on. So I'm more talking about the ion transport, this part. And this one we also develop, they can also develop, but I will skip at this time. So this is the famous scheme that probably about 2008, right? The Sterlinsis group, we started DNA sequencing, that we try to develop recognition tunneling. Then we can detect the moving molecule when it goes through the nanopore. But still, the moving molecule actually quite difficult to detect compared with the molecule immobilized on the surface. So here show that we can, using carbon tube, we can actually detect individual very small, like DCMP, so they're really small, about 1 nanometer. And also we can regulate how the molecule transports through. But to make those devices work, actually not easy, right? We fabricate those devices, but the yield is very low. So can we have a simpler way that we can detect those very small molecules when they're moving through the channel? So also from the carbon tube, actually we learned we can, using the DNA, when they're trapped inside of the carbon tube, they actually can filter the ion transport. So like DNA negative charge, so more positive ion will surround the DNA. And also we have significant ion concentration polarization happen there. So then basically the ion concentration not uniform everywhere. They become like really, you have a different concentration gradient across the carbon tube and also especially the entrance and the exit place. So we try to mimic that to enhance our sensitivity. So we use hydrogel put outside. In that way, we can slow down the small molecule transport. The thing is, we cannot make carbon tube that's small, like 2 nanometer. Then we try to use hydrogel, like fibrin hydrogel, normally used in the tissue, to slow down the molecule exit. So like here, the frozen image shows that really we can slow down the molecule movement when they're coming out. Then they become jammed inside. So when they jam inside, similar like a carbon tube trapped in the, similar like DNA trapped in the carbon tube, that we have a many small molecule trapped, not one, but many molecule can trap there. So in that way, actually, we get a very high sensitivity. We can detect the ATP, similar like the small DCMP detect in carbon tube. We can detect that about 1 nanometer ATP through more than 10 nanometer size nanotube, nanopipette. So I probably will skip here. So one thing that recently we also found we actually can detect the structure of the proton complex. Like here, the stratividin and the biotin, they bind. The biotin is really small, right? We can detect actually this multi-peak structure. The bigger peak from stratividin and a small peak due to the biotin molecule. So with the increased size of biotin, you derive the biotin molecule, we can see the smaller peak actually increase. So that reflects structure. That's one thing that actually due to the concentrating gradient we created when we crowded the molecule and the tube. So that's similar to the DNA, similar to the carbon nanotube cases. So then we can have that resolution, we can resolve the two particles, they connect to each other. So that's probably one way we actually can, so we learned from early work and ASU, so we can achieve a similar resolution by creating a significant iron concentrating gradient. So I will stop here. Those are the people working on the project and the funding agencies, and also thank you for your patience. So the two places, right, the Sonora Desert, and then now our FIU is near the Everglades swamp. Thank you.
Maybe that's what our Florida friend meant by draining the swamp. Anyway, Jim, thank you very much. Time for one quick question. Right, you haven't scared them all off. Okay, Josh.
So just real quick, have you thought about integrating the ramen with this nanopore, nanopipette system?
Yeah, I think about, but didn't started yet. So one thing we can actually pass through some gonad particle with some molecule, and the other side is the metal-coated pole, right? So you can get signal. But still thinking about, but didn't really started yet.
Seeing the swamp reminds me, Jim, that you got me as a present an alligator
Yeah.
for my 60th field trip.
Yeah.
Here, so. And I was also listening to your talk, pleased to be reminded that there was a time when the lab could do complicated experiments. I don't think we can do them anymore. Jim, thank you again.
Okay. Thank you.